Okay, last but not least, we're going to hear from Ron Weiss, who's Associate Professor of Biological Engineering, and he works on synthetic gene networks.
Okay, thank you for inviting me. A pleasure to be here. I'm actually a computer scientist by training, and over the last 15 years or so, I've been involved in synthetic biology, and probably about half of the time, the last half of that time, I've also been working on mammalian synthetic biology, and that's become more and more of a focus of what we do in my lab. And one of the areas that I'm interested in is how can mammalian synthetic biology help with cancer research and perhaps with cancer therapy. So I'm going to try to tell you a little bit about that and some of the kind of the basic steps that we're taking along that way. And so in synthetic biology, we often get inspired by other engineering disciplines where the notion is that you can take parts and put parts together and create modules, and then modules together to create systems, and these could be single entity systems or it could be multi-entity systems. And so we're asking in synthetic biology, can we do the same? Can we take proteins, create basic logic operations out of them, put them together into synthetic pathways that have desired functions, take those pathways, put them into the cells, and perhaps create programmable tissues and programmable aggregates of cells? And so that's what we're asking. In order to approach this, one of my fantasies, if you will, as a computer scientist, is that the bioengineers of the future will mostly be writing biocode, so genetic programs, something that looks like this. This is a little bit probably ugly to most of you. This is historical reasons. We use a lot of parentheses, but this is another way of looking at it. So this is a simple program that we can encode, which is if some condition, they make a cyan fluorescent protein, else different condition produce a yellow fluorescent protein. So this is a simple program that our biocompiler can take and then automatically translate that through a series of steps into genetic regulatory networks. So for example, this is a gene network that is based on parts that we actually have in the freezer that we've characterized. And then this biocompiler, not only that, it can also provide instructions for a liquid handling robot to actually do assembly in an automated fashion. So in my lab right now, the robot can do about 90% of the work. We actually, my former advisor, for example, has a company that most of the DNA assembly now happens automatically. And every once in a while, you need a person to move a plate around. And so this notion that graduate students and postdocs build DNA is going to be passé relatively soon, maybe in the next 2 to 5 years or so. So this is really becoming a reality, certainly easier than curing cancer. One of the things that we're working on is then taking this notion of a biocompiler and also applying it to create a biocompiler for cancer. In particular, take this notion of looking at databases of microRNA profiles and having the biocompiler automatically create a genetic circuit that can determine that particular cancer type based on the microRNA profile versus all other healthy cells. And so this is the notion. So we're attacking right now the challenge of specificity. Okay, so this is not the only challenge in cancer, but it is an important challenge, and it's related to other challenges as well. It's certainly related to delivery, for example. So if you can have something that's contains much higher specificity, then you can deliver, for example, a much higher dosage of your therapeutic agent. And so this is something, the specificity challenge, I think, is one that synthetic biology in particular can have a very nice approach to. And so in our approach, we do not have a one-to-one correlation between a biomarker and saying that this indicates that is a particular cancer cell type. So we believe that in most, if not all cases, that just doesn't exist. So kind of relating back to Doug's talk, we want to take a more system-level approach. So we want to be able to create these therapeutic agents that go inside the cell and then sense many elements, sense many biomarkers. And in this particular case, it's microRNA, but we're also looking at proteins. We're also looking at messenger RNA biomarkers, and then create logic functions that integrate multiple sensory information, combine that, and then come up with a single answer. Is it that particular cancer that we care about, or is it not? Okay, so kind of a notion of sensory inputs. It could be analog concentrations of biomarker levels, but mostly digital output, a yes or no output about cancer or not. And there are non-clinical applications that we're looking at. So, for example, if we want to do more sophisticated diagnostics, not go into clinical trials just yet, we believe that these multi-input logic functions coupled to sensors can have other interesting roles with respect to understanding whether it's development, whether cancer progression, and so on. And so we focused on Hela cells. Those are easier to deal with. And so the idea is that the cancer classifiers go into the cells, and they try to do the following: detect and kill Hela cells, and this is the bioprogram. Okay, so again, this notion of ugliness pervades this, but you'll have to forgive me for that. But this is a simple bioprogram. Okay, this basically just says these microRNAs have to be high. The ones on the bottom have to be, on the left have to be high. These have to be low. If that's the case, that's a Hela cell. Okay, and it's not a healthy cell. And we said, by looking at microRNA atlas data, we said that this appears to be a profile that really is indicative with high level of specificity to Hela cells. And so this is what it would look like. So we have a cancer classifier that's encapsulated, some kind of an agent that goes into the cells, does this logic operation, and then controls the expression of an apoptotic protein. Okay? This is a simple slide that I want to tell you. Basically, more is better. Okay? And so probably most of you will agree with it. When we looked at a few microRNA biomarkers, they were just not enough. They didn't give us the right level of specificity. But as we increase the number of biomarkers, we get higher and higher specificity. And we thought in this particular case that 5 or 6 would actually give us the right level of specificity. An important question then is, for other cancer types, what is the right level of specificity? So we don't have the answer to that. We are, with a biocompiler, hopefully going to get some computational predictions about that. And the hope is that for many cancer cell types, it would be a manageable number, whether it's 5, 10, 20 biomarkers, something that we can actually encode in a therapeutic agent. How do those work? So I'll give you a quick indication of how we actually built these systems, and then tested them. So the simplest sensor is a Hela low microRNA sensor. The idea is that the output is basically the knot of the input. Okay, so this is simple to do. So if the microRNA is high, we have target sites on the output. And so microRNA is high, that means we have no output, and vice versa. So this is implementing the knot operation. You can then make this slightly more complex by having basically an AND gate of NOT operations. And so the idea here is the output is high only when all inputs are low. So this is an AND of NOT operations. And then you can continue and you can create Hela high sensors. So the idea is that the output is high when the microRNA is high. Okay, and so these, for example, would be markers that would be high in Hela cells. They can be high in other cells as well, but they have to be high in Hela cells. So how does this work? This is basically a not operation. Okay, and this, I'm not going to go into all the details of how this works, but this is microRNA that basically represses a transcriptional activator, which activates a transcriptional repressor. Okay, again, not operation. You can then couple them to things that are now getting more and more sophisticated. So this is an AND operation of 2 Hela high sensors. Okay, and this is how you build something like this. Again, I won't go into the details of that, but it does appear to work. So we can combine this AND gate of 2 Hela high sensors with the AND gate of the 3 Hela low sensors. And so this is the final circuit that we built. So now you see that it's got a lot of interactive parts. Does this work? So in cell culture, this works. This is the circuit. Again, this is the representation. And these are experimental results in terms of Hela output that we get from the circuit. So this is a red fluorescent protein versus, now we're not looking at healthy cells. We're actually looking at immortalized cell lines. So these are actually cell lines that have cancer properties. And we're showing that the circuit can even distinguish between Hela cells and cells that are at least partially related. So now obviously, in a therapeutic setting, if we killed other cancer cells, the patients probably won't complain too much. But here, the emphasis on showing specificity. Okay, we can take this and now couple this to an apoptotic protein. In this particular case, a Bax protein, and we're showing that the Hela cells are mostly killed by the circuit, almost completely killed by the circuit relative to the positive control, and the worst behaving cells are the HEK cells, and they're mostly unaffected. So we have not only high level of specificity with respect to a fluorescent protein, but also when we mix these cells together in cell culture, we have a high level of specificity with respect to killing. And this was done simply with multiple plasmid co-transfection. Okay, and so we have started. This is looking more at the population level. We are now actively looking at the single cell behavior, and so these are the results in terms of fluorescence of the single cell behavior. And we are now doing optimizations to give us better single cell behavior. And so you see, by going from multi-plasmid co-transfection to single plasmid transfection. Okay, this is a relatively large plasma, but single plasmid, we are getting much better results with respect to, again, the worst behaving cells at the single cell level. Okay, so we're looking at the performance of the circuit at the single cell level. We're also now spending a lot of time in understanding single cell multi-biomarker correlations. And this, I think, is a really exciting area. So not just looking at kind of the homogeneous behavior of the population, but understanding single cell correlations, understanding, for example, what's the dominant population, what are also subpopulations. Could be 0.5% or lower subpopulations. And now we have active projects to look at that and then be able to identify logic circuits that would be able to recognize those, even if they're not the dominant populations in the area. So clearly, there are many steps that we need to take along the way to go to therapy. And also, as I mentioned, there are other opportunities to take this technology for cell type identification, whether it's cancer progression, so we can do live cell sensors to understand the progression over time without having to kill the cells. We have other activities, for example, for tissue engineering to use this kind of technology. And one thing that I briefly want to mention is another technology which I think is relevant with respect to actual potential clinical usage, which is something we've gotten really excited about recently. And this is the notion that what I showed you was done with DNA, but as you can imagine, we have the potential chromosomal integration. So if we're curing one cancer, we might actually create other kinds of cancer if we're dealing with DNA. And so here we're focusing on encoding all of these capabilities just with RNA. So deliver RNA to the cells, and everything happens with RNA and above. And in particular, in this case, with RNA binding proteins that encode all the power that we have with DNA. So just very briefly, so we've actually implemented the same kind of sensor using RNA-only circuits. We've implemented cascades and switches, and I just want to show you, even with one sensor, now this is results just with RNA. So we put in RNA. This is a single marker that can now distinguish between HEK and Hela cells. So it works quite well. We've also tested a 4-input circuit between HEK, MCF7, and Hela cells, and works quite well. Now we're testing the full 6-input circuits. We can build cascades that work quite well, that have really good on-off behavior. We have things like 1-bit memory switches that work really nicely just purely with RNA. So this is a technology that I'm really excited about, whether it's for RNA therapeutics or other kinds of therapies that we're thinking about for vaccination and potential cell fate determination and control. So in summary, we're building a variety of foundational technologies, such as some of the ones that we discussed here, and then trying to find particular areas where we think synthetic biology changes how you approach problems, from this notion of programmability. And so with that, lots of people to thank. Certainly I want to thank you as well.
Questions for this provocative talk? Yeah, please.
Technology adaptable to using various reporter assay or reporter constructs for the common transcriptional pathways that are activated in various cancers, in which the reporters would lead to the production of individual microRNAs so that the aggregate could be read out, similarly to the method that you just described.
Yeah, so we have various efforts, both in terms of the input, so varying the kinds of inputs that we have. So we have particular technologies for looking at transcriptional activation. So for example, if you have a promoter that gets activated in particular instances, we now have a project where we're able to read out promoter activity, connect that to the logic circuits, have fluorescent protein readouts, but also have regulatory readouts as well, regulatory activities. So be able to read something like transcriptional activation, but then be able to even activate any gene that we want. For example, with the Cas CRISPR technologies, so you can actually activate or down-regulate essentially anything that you want.
Could you then create a construct that would be selectively lethal for cancer cells that have high STAT3 but not NF-kappa B, high retinoblastoma activity or E2F1 activity, but not something else? In other words, create very specific conditions that use the microRNA technology.
Yes. That's definitely. We can talk about the details of how we would do that, but yes, basically yes.
So I have a question that has to do with subtle differences between cells.
Yes.
So I'm sure that Hela differs from the 10 or 20 different lines you have analyzed by microRNA expression. But in fact, let's take the position that there's at least one normal cell type out there that has enough of an overlap in its microRNA expression at some point in its life, in the right organ, at the right pH, in the right epigenetic configuration or whatever, that in fact it looks like Hela, looks enough like Hela that when the vector comes and the moment of truth comes, it too dies.
Right.
Have you thought about?
Yeah,
how
I mean
to deal with
this
that
is obviously a critical question, and so we are trying to approach that by first of all coming up with so if it has enough overlap, what does that mean? So we want to find situations where if it has 98% overlap in terms of its microRNA expression, we want to find where the differences are. And so I would argue that there has to be somewhere a difference. If the normal cell is completely identical to the disease cell, then they're the same. And
it's one parameter.
Yeah.
It's one parameter.
So the
relatively small parameter, the
Right.
number and distribution of a small number of microRNAs
Right.
compared to the total.
So the question is, can we find these specific not whether they exist, but can we find them and can we implement them and can we have profiles that work kind of in steady state situation, but also looking at the dynamics. So how do these profiles change over time? How do these profiles behave in an in vivo context? And so this is the question that we're looking at right now. Sure. Is can we understand variations? Can we understand the very subtle differences? Can we understand the progression over time? Yeah.
Okay, it's late. Okay, but it's not over. So the maybe a pretty good ending to this will be that all of our colleagues, hopefully from MIT, will join any of you who want outside that door for a drink and an hors d'oeuvre. And that's a chance for you all to get a chance to talk in more detail and without having to listen to me for another moment. I thank you all, every speaker.